The currency of the brain is electricity. Our sensory and motor abilities rely on the faithful relay of electrical signals both along and between neurons. We can think of this process in the same way that we think of communication along a telephone line. If cables aren't properly insulated, signals can be lost or distorted. Neuronal projections are ensheathed with a special insulating material known as myelin, and loss of this insulation can likewise lead to signal distortion. We see this manifest in demyelinating diseases, where oligodendrocytes, the cells that provide myelin, die. The subsequent loss of myelin causes devastating impairments, both in sensation and motor control. The good news here is that our brain has a population of progenitor cells that we can think of as teeny tiny cable repairmen. These oligodendrocyte progenitor cells, or OPCs, respond to myelin loss by migrating to lesion sites and maturing into oligodendrocytes to repair myelin and restore conduction. Unfortunately, in a majority of patients, we see unexplained deficiencies in this repair response. And this has prompted our lab, among others, to pursue the question, why does remyelination fail? We took a special interest in the influence of infectious environmental factors, notably herpes viruses. Herpes viruses are unique in their capacity to enter a state called latency, in which they can suppress their own replication to evade immune pressure. I'm going to guess that most people here have experienced chickenpox as a child, and that some may have even been unfortunate enough to see it reactivate as shingles. This is a perfect illustration of the herpes virus life cycle, with latency as a so-called benign state that lies between initial infection and reactivation. So, despite our mostly healthy appearances, all of us host a remarkable number of latent viruses, both peripherally and in our brains. Now, the idea that viruses could play a role in demyelinating disease is not new. In fact, it was first suggested back in the mid-19th century. However, our lab was the first to investigate the influence that latent herpes viruses might have not on myelin loss, but in the context of remyelination. In our studies, we looked at human herpes virus 6, or HHV6, which is the most commonly detected herpes virus in the brain, and has been clinically linked to demyelinating disease for over 20 years. We found that when we infect OPCs with the primary HHV6 latency gene, it significantly inhibited the migration of these cells when transplanted into a demyelinated mouse model. This was very surprising to us and suggested for the first time that latent herpes viruses may not be as benign as we previously thought. In fact, we propose that latent HHV6 present in the brain can impede white matter repair. It seems that this viral gene is making our little cable repairmen sick or injured, keeping them from getting to work. This leaves our neuronal cables uninsulated and subject to further degeneration. We're now hoping to figure out just how latent HHV6 could be inhibiting migration, with the hopes of identifying a novel therapeutic target to enhance remyelination and restore function in patients with demyelinating disease.